Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers, everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. We're thrilled by the overwhelming support for our episodes featuring top-notch experts delving into various biological realms. Thanks to the feedback from higher biology students, we're launching shorter, punchy revision episodes. Get ready for quick summaries of key areas to turbocharge your exam prep. These short episodes can support your exam preparation and are not intended to replace other important aspects of your revision. Before we dive into the genetic control of metabolism key area, let's start with a really important question. Why bother improving microorganisms in the first place? Well, wild strains of microorganisms, the types that you might find in nature, well, they can be useful. They can ferment sugars, produce antibiotics, or break down waste, but often they're not very efficient. By improving them using mutagenesis or recombinant DNA technology, we can increase the yield of valuable products like insulin or antibiotics, make microbes that grow faster or survive in tougher conditions, enable them to produce new substances such as human proteins, and reduce waste or byproducts in industrial processes. For example, E. coli can be genetically modified to produce human insulin, which is then harvested and purified to treat diabetes. Whether it's making biofuels, producing vaccines, or cleaning up oil spills, improving microorganisms lets us solve real-world problems more effectively and more sustainably. Now let's have a wee look at how this is done. Well, wild strains of microorganisms can be improved or increased in yield through mutagenesis or recombinant DNA technology. Mutagenesis is the creation of mutations. A mutation is a random change to DNA. While mutations are naturally rare, we can increase the chance of them happening by exposing organisms to mutagenic agents. Now, those include radiation like X-rays, gamma rays and UV light, as well as chemicals such as mustard gas. Now, here's something to keep in mind. Mutant strains are often genetically unstable. Over time, they tend to revert back to their original wild-type form, especially when grown continuously. Still, some mutations are beneficial and can produce improved strains of microorganism, like a yeast strain that ferments faster or bacteria that produce more antibiotic. Next, we've got recombinant DNA technology or genetic modification. Now, this is the deliberate transfer of genes from one strain or species to another unrelated one. For example, inserting a human insulin gene into bacteria so they can produce insulin. This process uses vectors, which are DNA molecules that carry foreign genes into another cell. These can be recombinant plasmids or artificial chromosomes. When we need to insert large fragments of DNA, artificial chromosomes are preferred over plasmids, like upgrading from a flash drive to an external hard drive, I guess. Now, we need to know the stages or the steps in recombinant DNA technology. It starts by identifying the desired gene. We then use a restriction endonuclease enzyme to remove that gene. We then remove a plasmid from a bacterium and cut it open with the same enzyme. We use DNA ligase to seal the gene into the plasmid, and we insert the recombinant plasmid into a bacterium before the desired protein is produced and then separated from the culture. These enzymes act like molecular scissors, cutting DNA at specific target sequences called restriction sites. Now, these sites are found on both DNA strands running in opposite directions. To ensure compatibility, the same restriction enzyme must be used to remove the desired gene from a chromosome and cut open the plasmid. That leaves sticky ends, short overhanging DNA sequences that help the gene stick to the plasmid. DNA ligase then joins or seals the gene into the plasmid or artificial chromosome, like using glue after cutting and aligning puzzle pieces. We also need to have an awareness of the structures of recombinant vectors. Now, recombinant plasmids and artificial chromosomes contain restriction sites where enzymes cut the DNA. They include regulatory sequences to control gene expression and an origin of replication so the DNA can replicate itself. They also have selectable markers such as antibiotic-resistant genes, which help identify transformed cells. For instance, only bacteria with a plasmid can grow on antibiotic-containing plates. And finally, they also have safety genes, which prevent the microbe from surviving outside the lab, a crucial biocontainment feature. Now, there is a bit of a catch. When we express plant or animal genes in bacteria, the resulting proteins are often folded incorrectly, making them inactive. That's because bacteria are prokaryotic and don't have the machinery to fold complex eukaryotic proteins correctly. So instead of bacteria, we often use recombinant yeast cells. Yeast is eukaryotic, like plants and animals, so it ensures the proteins are properly folded and active. A great example is using yeast to produce human insulin or vaccines, where correct folding is crucial for biological activity. So, we've explored why we improve microorganisms, to make them more efficient, productive, or capable of doing new and useful things. That can be done using mutagenesis, which introduces random mutations or recombinant DNA technology, which allows us to deliberately insert new genes. We've looked at how restriction enzymes and DNA ligases help us to build recombinant DNA, how plasmids and artificial chromosomes act as vectors, and what features they need, like replication origins and selectable markers. Finally, we've covered the limitations of bacteria in expressing complex eukaryotic genes and why yeast is often a better alternative for producing properly folded active proteins. So that's it for genetic control of metabolism and for Unit 2. So be sure to use these podcasts as part of your wider revision as you learn the key terms and processes and prepare for your SQA assessments. So hit subscribe on whatever platform you get your podcasts so you don't miss the rest of our short summary episodes nor our regular episodes with leading experts. And remember, this quick summary is intended as just one element of your broader revision plan. There are lots of resources available via Glow such as Scholar or WestOS, as well as BBC Bitesize. Or why not check out one of Lecky's great revision books and use the code LeckyPodcast40 to get 40% off on their website www.leckyscotland.co.uk.